Hello, everyone. Thank you for the presentation. I'm a PhD student at Lisbon University at Portugal. And today I'll be talking a bit about my work I've been developing for my PhD thesis that focused on the study of biophysical properties and organization of membrane domains and compartments in the fungal cells. And actually, fungal infections are a global overlooked threat, which incidence has been increasing. Annually, more than 1 billion people are affected by fungal infections, which results in approximately 1.7 million deaths. To fight this, it is imperative to characterize biophysically one of the main targets of antifungal drugs, that is the fungal plasma membrane. This will allow to better understand the interaction between these antifungal drugs and the plasma membrane, find new drug targets, and further develop or improve antifungal drugs already available in the market. And if we look at the eukaryotic membrane, typically, it is very heterogeneous, having different lipid domains with different biophysical properties that account for a plethora of functions in cells. And the fungal plasma membrane is not an exception. Particularly, the yeast plasma membrane has different lipid domains, such as the sterol-enriched domains or the sphingolipid-enriched domains, also known as SLEDs. And these SLEDs, interestingly, they are a feature of the yeast plasma membrane and of at least 2 other fungal species. In terms of composition, they are ergosterol depleted, and biophysically, they are highly ordered gel domains. And this makes sphingolipids potential molecular targets for new antifungal therapies. And actually, if we compare the sphingolipid structure of found in mammals with the ones found in fungi, they are different. For instance, inositol phosphorylceramides are a group of sphingolipids, complex sphingolipids found in the fungal plasma membrane that are absent in mammals, which makes them good candidates for enriching the SLEDs I just mentioned. Actually, in the plasma membrane of Saccharomyces cerevisiae, the most abundant of this complex sphingolipid is this MIP2C, the inositol phosphorylceramide. And in our lab, we have this IPT1 delta strain of the yeast that lacks this MIP2C and instead accumulates its precursor, MIP-C, that is manosyl inositol phosphorylceramide. And if you take a closer look, when we compare both of these sphingolipids, MIP-C lacks this inositol phosphate group, having a smaller polar head group and with a different charge. Interestingly, this IPT1 delta strain is resistant to some antifungal agents, as is the SCS7 delta strain that we also have in our lab. And in this strain, the complex sphingolipid found in the plasma membrane of the yeast lack the hydroxylation of the acyl chain in position 2, as shown here. I've been talking about membrane domains, but actually many proteins in the yeast plasma membrane are organized in these highly spatiotemporal stable membrane compartments, and particularly for the Saccharomyces plasma membrane, the 2 major non-overlapping membrane compartments are the MCC isosome and the MCP. The MCC contains the arginine permease CAN1, while the MCP has as a hallmark protein this ATPase PM1. And this PM1 protein is the most abundant protein found in the plasma membrane of fungi, having important functions such as creating and maintaining the transmembrane potential and regulating intracellular pH. While there is direct evidence for the MCC being ergosterol-enriched, there is only some evidence for the MCP being sphingolipid related, and only until recently there was direct evidence for this MCP, this membrane compartment, being sphingolipid enriched and co-localizing with SLEDs. And due to this, we decided to clarify the relationship that the MCC and the MCP have with SLEDs and ergosterol-enriched domains. To do so, we evaluated the role of the polar head group and the hydroxylation of sphingolipids in the plasma membrane biophysical properties, in particular of gel domains, sterol-enriched domains, and the 2 membrane compartments I mentioned, the MCC and the MCP. To achieve this goal, we used 3 different strains of Saccharomyces cerevisiae, the ones already mentioned, wild type, the IPT1 delta strain, and the SCS7 delta strain. And if you remember, in this first mutant strain, the IPT1 delta strain, the complex sphingolipids have a different head group when compared with wild type, while in the SCS7 delta strain, there is a different sphingolipid hydroxylation pattern. And for both mutant strains, there is no differences regarding sterol and glycerophospholipid composition when compared with the wild type. For these 3 strains, their membranes were labeled with different membrane fluorescent probes that partition preferentially towards different regions, different domains of the membrane, reporting on their biophysical properties. And these biophysical properties were studied using fluorescence spectroscopy and microscopy. So first, we characterized gel domains. To do so, we performed time-resolved fluorescence measurements of TPNA in living cells of the 3 strains. TPNA is transperinaric acid, a fluorescence membrane probe that has a strong preference towards gel domains. We obtained the fluorescence intensity decay for the probe when labeling the plasma membrane of each strain, and this decay is properly described by a multi-exponential model according to this equation, from which we obtain certain parameters that are required for the determination of the fluorescence lifetime of the probe, which actually reports on the hydrophobic chain packing of the gel domains. Also, one of these parameters, the long lifetime component of the probe, is a fingerprint for the presence of gel domains when longer than 30 nanoseconds. And if we look at the results we obtained, actually for the 3 strains, this long lifetime component was longer than 30 nanoseconds, confirming the presence of gel domains for the 3 strains. Also, the long lifetime component and the fluorescence lifetime was higher for both mutant strains when compared with the wild type. And this suggests that both mutant strains have gel domains with higher rigidity than the wild type. That is, that the sphingolipid alterations regarding the head group and the hydroxylation are affecting the properties of gel domains. And we can actually say that these gel domains are the SLEDs I mentioned at the beginning of the presentation. We also characterize sterol-enriched domains using fluorescence lifetime imaging microscopy, FLIM, of the 8-ANEPS. The 8-ANEPS probes sterol-enriched domains, and it is another fluorescent membrane probe. And in this technique, in FLIM, what we do is that we select a region of interest, a ROI, that is the plasma membrane for each cell, as you see in these figures, for each one of the strains in study. And then for each cell, for each one of these ROI, a fluorescence intensity decay is generated and analyzed, from which we obtained parameters important for the determination of the fluorescence lifetime of the probe, which is actually sensitive to the microenvironment of the membrane when surrounded by sterile molecules. And this fluorescence lifetime of the probe remained unchanged between the 3 strains, which suggests that the properties of sterol-enriched domains are not being affected by changes in the sphingolipid profile. Finally, we tagged CAN1 and PM1 respectively with GFP and MRFP, green and red fluorescence protein, in each one of the strains in study, and then performed fluorescence microscopy. And if you remember the beginning of the presentation, CAN1 is distributed along the compartment MCC that has been ergosterol related, while PM1 protein is a hallmark protein of the MCP that has been sphingolipid related. We performed FLIM for both tagged proteins to obtain the fluorescence lifetime that reports on the microenvironment surrounding each one of this protein. And also using the fluorescence images, we determined the heterogeneity distribution of each protein in the plasma membrane. And our results suggest that the change in sphingolipid profile did not affect significantly the distribution and surrounding environment of CAN1, while it did affect the distribution and surrounding environment of PM1. And if we take a closer look at all the results, we can see that the properties of gel domains that are actually SLEDs and the MCP were affected by changes in sphingolipid head group and hydroxylation, while neither ergosterol or ergosterol-enriched domains nor the MCC isosome properties were affected by these changes. And with that, we can conclude that this work reinforces the notion that SLEDs are distinct from ergosterol-enriched domains, also that MIP2C head group and acyl chain to hydroxylation are important for a proper hydrophobic chain packing of sphingolipids in the gel domains of wild-type cells. Interestingly, the increased rigidity of gel domains reported for both mutant strains could hinder the insertion of certain antifungal agents into the membrane, such as sphingomycin E, and justifies their higher resistance to this drug. Furthermore, and most importantly, together with some results recently published regarding the paralipidome of PM1E, PM1P having high sphingolipid content, our results suggest that SLEDs are enriched in the MCP. And just as a final note, last month, this article was published by a different group in which they used cryo-EM to study the conformation and organization of PM1 along the membrane, and also the lipids that the proteins surround. And the results in this paper fit perfectly with the conclusions that I just presented. And with that, I would just like to thank FCT for funding, to the organization of this contest for the opportunity to present my work, also to all the people that were important for the development of this work, particularly my supervisor, Dr. Rodrigo, and to all of you for listening. Thank you.